about the conserved breakpoints in disease. We'll be talking about the B chromosomes, and these are chromosomes that exist in variable number between animals in the same species, and they also are variable within an individual. So it should be obvious that this is a strange phenomenon. Also, I'll be talking about centromeric changes. And I have 4 hypotheses that I'll be talking about today. The first involves the divergent chromosome structures and the fact that regions of homologous sequence called evolutionarily conserved chromosome segments are reorganized in the Canidae. Also, as predicted in the fragile breakage model, these evolutionarily conserved chromosome segments, basically centenic regions, are common across the Canidae, and they're separated by evolutionary breakpoints. And these regions overlap in the Canidae. The third hypothesis revolves around the B chromosomes. And as I said before, these chromosomes exist in variable number in opposition to the A chromosomes, which are autosomes and sex chromosomes. We will show that the B chromosomes contain sequence that are found in the A chromosomes in addition to B chromosomes. And we believe that the B chromosomes arose intraspecifically, so within the species. Fourth hypothesis is that we believe that the rapid karyotypic restructuring that exists in Canidae, the rapid restructuring resulted in new chromosomes rising, and so new centromeres were created. And we believe that the Canidae is a great group to understand the centromerization cycle. So first I'll be talking about the 2 hypotheses 1 and 2. To address these 2 hypotheses, we utilize specific DNA sequence labeled as probes. And we labeled these as color-coded panels to represent different evolutionarily conserved segments. These were applied to the 12 canid species that I examined, and we identified recurrent breakpoint regions using these panels. We then compared these evolutionary breakpoint regions to regions of instability in canine cancer. So just a little bit of background of one of the major techniques that I used. This technique is called fluorescence in situ hybridization. Essentially, what you do with fish is that you will use a sequence, shown here, of interest, and this is labeled with usually a fluorescent tag, sometimes a hapten tag. And then it is applied to metaphase chromosomes. Excuse me. And in effect, what you're doing is anchoring sequence to physical places on the chromosome. So you could choose gene sequences and label them and apply them to chromosomes to identify the region of the chromosome where that lies. The other technique that I'll be talking briefly about today is array painting. And this technique utilizes flow-sorted chromosomes or chromosomes that are microdissected from cells and labels it with, again, a fluorophore, or labels it with a fluorophore, and then that is applied to microarrays. And the result is that you can identify sequence that is present on that chromosome. So what I'm showing you here is an example of these chromosome panels where I've labeled each region, each sequence that I'm interested in to represent dog chromosome 1. So what you should be able to see, and I'm not sure how easy it is to read, the first is red, then blue, then yellow, and purple, and so on. And so the pattern of these colors will enable me to identify not only the region of chromosome that these lie on, but also orient those sections. So in this example, chromosome 1 probe panel was applied to dog. So this is dog metaphase chromosome spreads. So what you should be able to see is that here and here is where these chromosome panels have hybridized. And with the way that we've designed this, you can actually look at the order of the colors. So here you see red, then blue, then orange, and so on, down to the bottom where there is blue and yellow. So you're able to not only identify the chromosome region where this lies, but also the orientation of the segment. And this is important because previous techniques